connective tissue. So definition for connective tissue is that it provides a mechanical function for structural support for tissues, organs, and bodies. So that helps maintain integrity and forms the body, organ, and the tissue. So the function of the connective tissues are to support defense mechanisms, are like for physical, immunologic, repair, storage, and transport. So then there's the connective components of the connective tissue. There's three parts of it. So cells, fibers, ground substance. So ground substance is like the extracellular matrix of the connective tissue. And then of the three components, they're broken up into like other subgroups. So we're gonna go first with cells. The cells of the connective tissue, there's two classes, either fixed or wandering. And then those are the examples of it, and then we're gonna go through that. So then fibers, there is collagen fiber, reticular fiber, and elastic fibers. And then we'll also go through examples of that. The next slide shows the collagen synthesis and structures. So there's a precursor alpha chain in the front. So there's three of them that combines together that makes the procollagen. And then, so this is happening in the ER lumen. And then once it's secreted out of the lumen, the ends are cut off. And I believe, so then it's collagen molecules, which then gets assembled into collagen fibril, which is the one that looks like, I don't know, wall. So it's combined together, it's like staggered, so that, or I guess like for more strength. And then in between those staggers, the holes in there, the small gaps are measured to be 64 nanometers, separate from each other from the head to tail. And then, yeah, and so then it makes the collagen fiber. And then that's just an image to show like how, the size of it and how it's made. So then it's tropocollagen helix, and then it goes into microfibril, and then fibril, and then fibers. And then those are the image of the fibers.
Do we have to know that enzyme in the other slide? If you go back a slide, on the bottom right it says lysyl oxidase.
Oh, yes. The lysyl oxidase is linking the tropocollagen molecules. Yeah, and then so those are separated into either dense irregular and dense regular. And so I guess we just need to know like how it looks like. Okay, and so now we're going to get into elastic fiber. So there's three types of fibers. There's oxytalin, ileoquin, elastin. And so I believe so elastic fiber produces the fibroblasts and smooth muscle cells, composed of four elastins surrounded by a network of glycoprotein microfibrils, and then which is micromodulin 1 and 2, and then large molecule fibrillin forming I can't even pronounce that. Illusion. Okay. And then these are like the two amino acids that are elastins has same composition like collagen, but has two amino acids, desmosine and isodesmosine, which makes the elastin rubbery. Elastin also occurs in a fenestrated sheet form, so now we're going to go into the ground substance. So ground substance is a gel-like substance, resists force of compression and helps rapid change of nutrients and waste. It's made up of glycosaminoglycan, protoglycan, glycoprotein, it's also known as adhesive, and then others, protein, lipid, carbohydrates, and water. So the glycosaminoglycans, also known as GAGs, it's negatively charged polysaccharides, and which is repeats of disaccharides that bind to water. So one of the disaccharides is amino sugar, hexoamine, which is made of glycosamine or galactosamine. And then, so it's used, so since it's negatively charged, it can attract cations, and then, which makes up like the extracellular fluid for like hydration and also resistance of like compression. And then since it's negative, it repels off one another and causes a slippery feeling. And then most of the GAGs are sulfated, and it's bound to proteins to form protoglycans. And then the protoglycans contain a core protein that attaches with the GAGs attached to, and it's called like bottle brushes. I guess it looks like a bottle brush. It's connected to a hyaluronic acid by the protein core, and then connection reinforces small linkage proteins. And then so there's actually 3 types of protoglycans, and this is one of them, agrican. So it's presented in cartilage. And then these are the other 2, and it is, I guess, embedded in the epithelium. Okay. And cell surface matrix receptor. I was a little bit confused on this.
Can you go back one slide? So this syndecan, or the fibroglycan, it's attached to the surface epithelium. And you're saying this is for receptors, and that's why the next slide is related to this? Is that right? I don't know.
Oh, I was a little confused on the cell surface matrix receptor.
It says serves, so they serve to link the ECM to the intracellular actin filament through these complexes.
I'm just trying to find the connection to that with the GAGs.
All I remember him hearing was saying something about taurine and vinculin anchoring something, but I couldn't like understand it.
So those are, maybe they interact with those type of protoglycans, because those are the ones that are at the surface, right?
Yeah.
And they're just talking about what type of receptors, what are mediating them. They work with actin filament. I'm confused.
Yeah.
But then the next one is the last type of.
Yeah, the adhesion one. So these glycoproteins is not only connected to the cells, to extracellular matrix, but also guide their movements. Fibronectin, cell adhesion and migration. So that helps with migration, and laminin interact with cell receptor integrin, loosely connect the cells to basal lamina.
Oh, I think the significance of laminin is because it integrates with that.
Integrin.
integrin, and that integrin interacts with the taxin and the talin, paxillin, and vinculin.
Yeah.
So
basically, the integrin is a receptor on the cell surface, or sorry, the cell surface of the matrix receptor, and that's on laminin, which is a type of glycoprotein.
Cool. Well, thanks. Okay, and then we're going to go into fixed cells of the connective tissue. So we have mesenchymal cells. So precursor of most indigenous cells of the CT, loose network of satellite cells and abundant intracellular fluid. Some cells remain undifferentiated in adult tissue, difference to distinguish from fibroblasts. So fibroblasts are large, flat, and branched. They look like stars and spindle shape. So nucleus is oval and elongated, so it has 1 to 2 nuclei. So you can see there it's like kind of long. Young cells in cytoplasmic looks the same. And then reticular cells. Reticular cells are satellite cells with long, thin cytoplasmic processes, centrally located pale nucleus and prominent nucleolus. Produces reticular fiber that forms stoma of hematopoietic and lymphoid tissue. Some may be phagocytes, antigenic materials. And then other APC collects antigens on their surface to attract immune cells. Then we have adipose tissue. There's white and brown. So we're going to do the white one. It's loose connective tissue. It's unilocular, single large fat droplets, find everywhere, single or as a group, and signet ring cells. And then brown fat is considered a considerable amount of cytoplasm, multi-fat droplets of variable sizes, round nucleus, and almost centrally located. Lots of mitochondria prominent in newborns and scarce in adults. Okay, and the wandering cells. So we have mast cells. They're derived from marrows, abundant in basophilic granules. The granules contain heparin, histamine, and eosinophilic chemotactic factors of anaphylaxis, right? ECFA, which is also known for that short term. All right. And then we have our plasma cells, similar to lymphocytes, but more cytoplasm and eccentric nucleus. Nucleus large clump periphery located. Adjacent to nucleus is the Golgi apparatus, rare in connective tissues, secretes immunoglobulin antibodies. Cytoplasm have acetophilic granules known as Russell bodies. And then there are others.
Also, he emphasized this in class. I don't remember the specific parts, but he said if it looks like crescent shape, then you'll be able to figure out that, okay, it's plasma cell.